everyone who hugs Nathaniel looks tiny.
He sure smells good though.
That's good. I showered today.
No, I just said that about Ryan.
My hand, I feel like there's like an infinitely flat. I'm nude with that mug. All right. Okay, so I still haven't tabulated the latest points, but I have them. I have pictures. I have a video of them. So I'll add in the latest points. But if anyone can figure out who these two people are, here's the clue. I'm hungry. Then that team gets a half a bonus point if you can figure out who those two people are. Not right now, but whoever emails me first, because I have to have a timestamp of who said it first, unless you can just shout it now. I assume nobody in the room but me knows them, knows of them. As a hint, he's still alive.
Oh,
You just
that's as well. Okay.
take a picture.
Let me see it.
Yep, go for it.
Yep. I got you.
All right.
Figure it out.
So these are the points like 3 lectures ago. There are updates. I'll put them in. I will put them in so that on whatever it's Friday, on Monday, we'll have the updated scores. I think Team 3 still has the Mario star. I think they're still winning, but it's probably a race. It's probably a race. We'll keep going. Team names. Have we?
We doing jamming?
No, it's pronounced pajama.
Jama.
Yes.
And they have the beat with it. So team names, everybody, the cleverest, funniest, whateverest one gets a little bit of bonus points. Remember, here is where you can find all the PDFs. If there's a PDF in the slides, you'll find it there. And I'm going to put the review slides up this weekend because I've taken a lot of them down. Right, we're starting on lecture 7. The first 6, I'll have that review slide block up there. We can just download it on PDF for studying. You know the body type stuff. You know the problem of what healthy, obese, and skinny fat, how BMI doesn't quite characterize if you're Dwayne Johnson, if you're Princess Diana. BMI doesn't quite characterize where you fall. Remind me what metabolism is.
Chemical reaction.
Good, yeah, whatever, that stuff. What's this?
ATP.
ATP, what's it for?
ATP hydrolysis. ATP plus water. ATPase. ADP, PI, hydrogen ions.
Good. Yeah. Cameron, what are you 2? What are you 3?
Teacher's pad.
0.017. And the other ways, right? So the creatine kinase reaction, we have the adenylate kinase reaction, other ways to get our ATP back. We have spent it, ATP hydrolysis, we have to get it back. Glycolysis, that's another way. We'll talk a lot more about this later, but remember that's from glucose with hexokinase. If you're exercising, if you're a human being and you're not like yeast on the march, right? If you're a human being and you're exercising, hexokinase is not step 1. You'll do a little bit of it. Lipolysis, let's do it one more time, beginning to end. Okay, lipolysis, how does that work? Beta adrenergic receptor. What binds to that?
Epi, norepi.
Epi, norepi. What's the difference?
One.
Yeah, let's what team are you? 1.016. Cameron beat you. So norepi, that's coming from nerves. Remember, fat is innervated. Fat is not just a endocrine response for burning. You can get beta adrenergic stimulation with norepi or with epi. So you're coming from either the adrenal medulla or the nerves themselves. Fat is innervated. So what enzyme gets turned on? Adenylate cyclase. What does that do? What does that do? What does that do? So hormone-sensitive lipase, pyrrolipin. Remember with fat, the first one is adipose triglyceride lipase. And PKA does stimulate that a little bit, but when you look at the literature, it's like 13 times as much. It's stimulating hormone-sensitive lipase. It's really sensitive, hormone-sensitive lipases to PKA. And then pyrrolipin, what's that? Yeah, if I'm gonna like unbutton and stuff. Yeah, pyrrolipin is a co-protein. Another 0.015. So now we're mobilizing our fat. Adipose triglyceride lipase, hormone-sensitive lipase, and then monoacylglycerol lipase. So we've mobilized all three fatty acids from a triacylglycerol to a diacylglycerol to a mono, a triglyceride to a di to a mono. We get it into the blood. What do those fatty acids look like? When they're a triglyceride, the glycerol backbone, and then when we mobilize them, how many carbons? 16, 18 usually. It can be something else. It could even be odd. That's rare, but 16 to 18, that's almost all of them. 16 to 18. So then we get them to the metabolically active tissue. This is a peripheral adipocyte. We're going to send it to the metabolically active tissue. What happens to it there? It needs to be shuttled in. So we have whatever, a cell. Inside of the cell, we're going to have a mitochondria, a bunch. Yeah, so we have a transporter protein that's going to get it in, and we're going to put a coenzyme A on it. So we have an acyl CoA, and then bind it with carnitine. CPT1, let's put carnitine on it. So now we have an acyl carnitine. What does acyl carnitine do?
Into the cell.
Carnitine, acyl, carnitine,
Lipase. Yeah, so it's CACT, carnitine, acyl carnitine translocation. Why is both carnitine and acyl carnitine on there? CACT.
Carnitine has to go back out.
Because carnitine goes back out, free carnitine goes out, acyl carnitine comes in. So now in the mitochondrion we have a acyl carnitine, and we ship the free carnitine back out. You're two. So we now have acyl carnitine inside of a mitochondrion. The free carnitine has gone back out. What do we do with the acyl carnitine? Yeah, first we do that almost immediately. But first, what does CPT1 do? Yeah, CPT2. Get rid of the carnitine. So the carnitine can go back out. It's done its ushering duties. It can go usher in another acyl CoA. And so we remove the carnitine, so we get acyl CoA again. And then, Alex?
It gets dehydrogenated and oxidated.
Yeah, dehydrogenation and oxidation for that first step, and then? And then hydrated, and then? Dehydrogenase again, and then?
And then theolytic cleavage.
Yeah, theolytic cleavage. And so we're going to chop off a little 2-carbon chunk. So if it was 18, we now have a 16-carbon acyl CoA, and we have a 2-carbon feeder. Not the keto. If you combine them, acetyl CoA, if you combine them, you're going to get ketones. So the main way to get ketones is if you don't have oxaloacetate for the Krebs cycle in the liver, those acetyl CoAs get bound together. But we chop off a 2-carbon acetyl CoA. We have our acyl CoA that's going to keep undergoing beta oxidation. So if we have a 16-carbon fatty acid, yeah, 8. What if we have a 15?
Oh, that's 6.
Yeah. So if we go from, if it's 16 and we get 8, it'd be 7. Then the propanol we would get, and then it would go into the Krebs cycle eventually as succinyl CoA. And so we get a Krebs cycle intermediate. So we now have a bunch of acetyl CoAs that we've made through beta oxidation. What do we do with those acetyl CoAs? Krebs? They bind to oxaloacetate to make
Citrate.
Citrate undergoes a bunch of changes. We get oxaloacetate back. Those other two carbons, those releases carbon dioxide. What do we make in the Krebs cycle?
NADH, FADH2.
Yeah, one of those and three of the former. Three of the NADHs. And what are those things?
Coenzyme.
Electron transport chain.
Yeah, for the electron transport chain, they're donors. They're electron donors. Where does NADH donate?
Complex.
One.
Complex 1. Where's FADH2 donate? Complex 2. What complexes pump protons into that intermembrane space?
1 and 2.
1, 3,
1, 3,
and 4.
and 4. Yeah, 1, 3, and 4. And so which one is a little bit more potent in terms of its gradient, in terms of that proton gradient? 1, or NADH because of 1, because you get 4 protons pumped at complex 1. What is complex 4 called?
ATP.
Oh, yeah, cytochrome C oxidase. The ATP synthase is the enzyme. Oxidative phosphorylation at the end, that's ATP synthase. But cytochrome C oxidase, what inhibits it?
Cyanide.
Cyanide. Alison, what team are you?
3.
He's on fire.
0.022. So yeah, cyanide. So that's like the Appleseed amygdala and stuff.
That's what's removing the electrons.
The electrons will go eventually bind to oxygen. A couple of protons, a couple of electrons, part of oxygen where binds to. But cytochrome C oxidase is the enzyme that does it. That binds it. So we're good with lipolysis, right? Here's your triglyceride. The glycerol itself, you go through lipolysis, right? And so you're going to chop off all 3. You're going to lyse all 3 of those. The glycerol itself will undergo changes. That has some fates. Glycerol kinase. Eventually, you're going to get glyceraldehyde 3-phosphate. That can be used for gluconeogenesis. That's an intermediate in glycolysis. So glycerol itself has some fates. Insulin, tell me what insulin does to this.
Inhibits lipolysis because of PDE.
Yeah, it's going to activate PDE3B. That's the fat one. So it's phosphodiesterase. Insulin's going to activate phosphodiesterase, PI3K. PKB phosphodiesterase is going to activate it. Phosphodiesterase converts cyclic AMP into AMP. AMP doesn't activate PKA, so you don't get the downstream stuff. What else does insulin do that's more long term?
Reduce expression of adipose triglyceride lipase.
Perfect. Reduce the expression of adipose triglyceride lipase. Remember, that's the first step in lipolysis. And so long-term insulin is going to deplete the ability to do the first step in lipolysis, and directly it's going to inhibit phosphorylation of perilipin and activation of hormone-sensitive lipase. So this is we just went through all of this, right? And we'll mention carnitine again, but this idea of carnitine as an usher, taking those acyl-CoAs into a mitochondrion, because it can't just go in there on its own. It has to be escorted. It has to be ushered in there. So remember the CPT1 and 2, that's attach carnitine, remove carnitine. CPT1 and 2, attach, remove. CACT, as Nathaniel said, carnitine, acylcarnitine translocase. So you remove carnitine, you take it out of the mitochondrion, and you put acylcarnitine in, so you can keep doing it. So carnitine comes in, ushers, takes it to its beta oxidation seat, and then you remove that carnitine and it comes back out to usher in a new attendant. Just the steps, Alex went over these, the dehydration, hydration, dehydrogenation, hydration, dehydrogenation, theolytic cleavage, the lysate. Oh, it's the exact same thing, just a diagram of what you're getting. The acetyl-CoA, that's the feeder into the Krebs cycle. And then the acyl-CoA, let's keep chopping off little 2-carbon chunks. And remember, an odd carbon one, you're going to make succinyl-CoA out of it. And succinate, as people often say succinate, but succinate, either way, whatever the pronunciation, that we'll talk about a little bit more in just a second. But that acetyl-CoA comes into the Krebs cycle, binds to oxaloacetate, you get your citrate, undergoes changes, carbon dioxide, that's those 2 carbons, and you get all that NADH and FADH too. Those donate, right? Those donate to the electron transport chain, and oxidative phosphorylation includes ATP synthase. The electron transport chain, 1, 2, 3, 4. There's FADH, there's NADH. 1 and 2. 1, 3, and 4 pump the protons. And then at the end of that, we have this hydrogen ion, this proton gradient, and that drives ATP synthase to reconstitute your ATP. You remember the cyanide thing. So orange is the new black. What does arsenic actually inhibit? Pyruvate dehydrogenase. Fire. Okay, so remember the basal metabolism. We have a really difficult time controlling this. Exercise, thermogenesis. This is so close to nothing in our daily expense of calories. The thermic effect of food, typically more, depends what you're eating, right? It depends on your behaviors, what you're eating, how much you exercise, how little you eat, what you eat, whatever. But exercise is nearly nothing. Thermic effect of food tends to be a little bit more. NEAT, non-exercise activity thermogenesis, that's pretty big. That's pretty big. James Levine, he's at Mayo Clinic, and he looks at non-exercise activity thermogenesis, and he did what's called the magic underwear study, they call it, and he found that NEAT was one of the biggest explanations for people who are given excess calories and who doesn't develop adiposity, doesn't increase their BMI. So NEAT is important, but basal metabolic rate, man, that thing is huge. And it's based on your organs, the brain is doing a ton, ion gradients, temperature regulation, all of that stuff. So just remember the calories in, you can totally control that. You can measure stuff. You can see how many calories are on the label of what you're eating. Calories out, that changes drastically based on calories in. So it's a hard to control equation from just the laws of physics. Calories in, calories out makes sense. From a practical application, controlling calories out becomes really problematic because that just changes against your will without your input. Remember, fat is an endocrine organ. Fat secretes stuff. And so there's a lot of peptides it's releasing. Adiponectin, when you see the Golgi apparatus, right? This is the adipocyte. See Golgi apparatus, I think adiponectin is going through that. You know what mitochondria are, you know what a cytoplasm is. Here's where your triglycerides are, where lipolytic activity is going to happen. But fat, there are receptors all over it. There's receptors all over fat. It receives information from your endocrine system and it releases stuff at the same time. Just remember, you don't want to get too lean. Everybody, if you ask people, summer's coming up or whatever, and everyone wants to get like lean, like, well, how lean do you want? Ripped, as lean as I can get. Health consequences, right? There's potential health consequences. Guys can get into the single digits and be bitchy, I guess. When you get really low in body composition, when I did competitive bodybuilding in season, I would have to quit my job because I would be fired from my emotional outbursts if I kept my job. And so, and I mean, that's once you get down to 7 or so, life doesn't treat you well. And you ask a bunch of guys, what's your body fat percent? 7 is a magic number that everyone says when they're 25. Well, what's your body comp? 7. What's with the tummy then? So 7 is what everyone says. That's an unpleasant place to be. Nobody wants to actually exist at 7 other than have the mirror applaud you. For women, you get down to 7, you have real problems. 11, 12, 13, that's about the bottom that you'd want to go. Now, the speed. So talking about the rate of fat loss. If you're doing more than a pound or 2 a week, you're going to have complications. You're going to have a metabolic problem and overcoming a basal metabolic rate that is not agreeable, a basal metabolic rate that has no interest in working with you, has no interest in shedding additional pounds. That's hard to overcome. And so when we see these biggest loser competitors and their basal metabolism is horrible and they're doing more exercise than they ever have, their diets are on and they gain all their weight back. So you have to be cautious in pacing it. Now remember, there's different kinds of fat. The white fat, that's what we think of. White fat, especially with adults. With little kids, more brown fat. Historically, we think kids have a bunch of brown fat, but it's gone by the time you're adult. Not true. We have brown fat. Your grandparents have brown fat. Not as much as a little kid. And we'll talk about shivering. Kids don't shiver. And so thermal generation, they have other methods of this. And the brown fat is a huge method. But we also have beige fat. And you can convert the white into beige. Convert the white into beige. People are still looking up the mechanisms of that. Lactate is one of the triggers. There's multiple triggers for this. But brown fat, remember, tons of mitochondria in brown fat. Not so in white. Just the roles that they play. You don't really need to know the roles that they play, but it's a little bit different. And the visceral fat, that's the dangerous stuff. The general locations of where this stuff resides, which looks like little, I don't know, kidneys or something all over the body. This is the brown fat. In an infant, it's minimized a little bit. In a young adult, minimized a little bit more. And granddaddy and grandmommy, it's a little bit less, as visceral adiposity tends to increase. But we still have brown fat, and we can convert white into beige. And browning, let's get some beige adipocytes, more thermogenesis, better insulin sensitivity. Poor insulin sensitivity, that's terrible. Better insulin sensitivity, less of an inflammatory response, and less of an association with metabolic disorders when you're looking at the beige and the brown fat, as opposed to white fat, terrible with thermogenesis, worse insulin sensitivity, increased inflammation, increased association with metabolic disorders. So getting brown, getting beige, giving your fat a tan, that's helpful. Now, the ways to burn it, there are ways to burn it. Remember cold exposure. Just go be really, really cold for a little bit. And shiver. You have to shiver. If you're not shivering, you don't get the release of succinate, or again, like succinate sometimes it's called. You don't get the release of that. So you have to shiver to get the full effect. But epi, norepi, those increase with cold exposure, so you're going to get some beta-adrenergic signaling. Epi, norepi, those are going to increase with cold exposure. Norepi, remember, fat is innervated. And so you're going to see something close to submaximal exercise with a cold effect. Now, if you're shivering, you can add this effect to it. The muscle, you'll release this. It's an intermediate in the Krebs cycle. You'll release this with exercise, but cold exposure, shivering, you're going to release a bunch of this. Your fat will take it up. The brown fat is going to take it up. You know how, why are trees big? Born that way. They sequester all that carbon, right? So it's like, okay, here's like a bunch of carbon dioxide, and let's keep some of that carbon and convert it into bark and stuff, whatever. Your brown fat can sequester some of this succinate or succinate, but shiver, go be cold, but shiver, and your muscle will release this. The brown fat takes it up, very metabolically active. You're going to do a bunch of thermogenesis that way, and you can match a cardiovascular. Treadmills. Do you know why those were invented? The reason? What?
Pollution.
Cool. Yeah, they powered mills. That's the original aim. When they were powering mills. So, not quite, but close. Close. It was to torture prisoners. Look up the history of treadmills. It's fascinating. Oscar Wilde was put on a treadmill when he was in prison. He did walk. It was like a big step mill. A big at the back of those boats that sort of feel like you have lunch on them and they go a mile an hour down a river.
The paddle boats.
It's this huge paddle boats, a big paddle on the back. That's what the treadmills were. They had this just row of prisoners, 6 hours a day on sort of a stepper. And it was essentially for torture. And then it was banned. It was legally banned for cruel punishment. Somewhere around the turn of the 20th century. It was late 19th century is when they were in full effect, and that's when Oscar Wilde was tortured by treadmill. And then the patent started coming out of let's do this voluntarily. Let's do treadmills voluntarily. So it's okay if you don't like the treadmill. They were literally designed as a corrective, emotionally abusing machine. And it's okay if you don't like that, but just go be cold then. Go be cold, shiver a little bit. Don't give yourself frostbite. Don't give yourself hypothermia. Just get in cold water, get out and shiver. Get back in the cold water, get back out and shiver, and cycle a few times. Don't dry off, cycle a few times. The muscle will release that succinate or succinate. And
So you were mentioning, I think last class, about converting white fat to base fat using that method.
yeah, cold exposure does it.
Okay.
Yeah, exercise too. Cold exposure is the thing that I've seen in the literature
And they're
most.
like, we can recruit more if we just convert it. Kind of
I
thing?
think, yeah, it's the research is still relatively young there.
Okay.
And so when you look at some of the dates on the articles, 2018, and this is the first study that was looking at this, the first ever one. And it was 2008, the first one I'm aware of. And that was 2018. And starting to look at the benefits of cold exposure. This is a 2021 article. So this is all new stuff. And so they don't quite know all of the mechanisms, how it works. Exercise does it a little bit. Cold exposure is what I've seen in the research the most.
But is your assumption, like, I mean, strictly assumption, hypothesis, but it's like specificity adaptation kind of
Yeah, I like that.
reasoning? Yeah.
Yeah, specificity adaptation. We need that's really good. Yeah, we need thermogenesis. White fat doesn't do that.
Yeah.
Yeah, nice. That's the most points of the day. You're 2.036. That was good. I mean, I don't know if it beats Alex's collection of like 20 things,
Yeah.
but it did on the board. I'm sorry. But so when we start looking at cold exposure, there's a lot of things. Increase adiponectin, that's going to improve insulin sensitivity. Fat releases that adipocyte, adiponectin. Decrease leptin, increase lipolysis and fat oxidation. There's a lot of effects of cold exposure. You do build up a tolerance. So if you're living in the Antarctic somewhere, you're like, well, cold exposure every day. I joined an Eskimo tribe. I live in the igloo or whatever. You adapt, giving more credence to what Nathaniel just said. So optimizing fat oxidation through exercise and diet. Remember when the treadmill says that silly thing about here's the fat burning zone. There is research. There is research to the fat burning zone and comparing different people. It's not just like, what's it? It's like Woodway and Life Fitness and stuff, just making shit up. Sometimes they do. But there's research here. So 59 to 64%. There's a range for people who are more fit where they can oxidize fat. It also depends on diet, history of training, nutrition. All of these things are going to affect what the intensity of exercise is, where you can peak at fat oxidation. And then for unfit people who you just you go to like a coffee shop and people are just sitting there and they don't look like they do much else but sit, there's going to be a lower percentage of their VO2 max as an indication of intensity, a lower percentage. But for people who are much more fit, and so here's what we see as the intensity increases. This is we're increasing VO2 max. We're getting harder and harder and harder. We're pushing farther and farther. We are breathing harder and harder. We can barely go. And fat metabolism is eventually going to crash. Like there's none. But if you're highly trained, you can go a little bit farther and still have a high percent of fat oxidation, as well as absolute amount of fat oxidation. This is absolute amount of fat oxidation. Males and females, and so actually males are a lower and females are higher. Did I say it backwards on Wednesday? I have no idea what I said on Wednesday. The percentage of VO2 max and fat oxidation, females are a little bit higher. And then this is just the two different articles. Humans doing endurance exercise, looking at the source of fat. So starting with this one that just looks at plasma fatty acids, meaning peripheral, right? You're on the cardio, whatever it is, you're like the prison treadmill, and you're taking fat from your abs or something, peripheral, right? Plasma fatty acids, 25% exercise intensity, 25% of VO2 max, peripheral adipocytes. That's the big contributor. Intramuscular triglycerides, local stuff, very little. Increase the intensity to 65%. There's more total fatty acid consumption, but technically the peripheral stuff has gone down. You're relying a little bit more on the local fat. increase it even more, and both of them go down. The absolute amount of fat metabolism decreases when you go from 65 to 85. It continues to decrease with peripheral adipocytes, and it maxes out somewhere around here with local adipocytes, the intermuscular stuff, intermuscular trigger, triacylglycerol or triglyceride. Now this, you might go through lipolysis in those peripheral cells. You don't release them. You don't actually make it to the blood. After exercise, you might dump a few more. The rate of release of those fatty acids might go up after exercise, but you then tend to see something sort of similar in the total rate of fat metabolism, so maybe a little bit higher in the people who went extra hard. But you'll increase here, but then this also falls as you continue to increase because you compromise carnitine's ability to deliver those acyl-CoAs into a mitochondrion. So the mitochondria never see the fatty acids. So even the local stuff goes down. And so then what we see taking it, the bulk of all this, muscle glycogen is really what's going to do it. Muscle glycogen is like nothing at 25%. You just, who cares? You're not even going to use it. 65% you're using a fair amount. 85% glycogen is really what's doing it. And what happens, this is why you get this. The fat's there. This has a problem delivering fat. You don't release it out of the peripheral adipocytes. It just can't go there. You can't get out. It's sort of imprisoned. But this gets contracted because of this. You make all that acetyl-CoA out of glycolysis, and it blocks carnitine's ability to take fat into the mitochondria. So this is just one of the articles. There's a couple of articles. This is one of them talking about making all of that acetyl-CoA and trapping the carnitine so carnitine can't do its ushering of fatty acids. Now, keto adapted, what do we see? For keto adapted people, what do we see in terms of lipolysis? What do we see in energy substrate use? So this is peak oxidation. So how much fat can you consume? This is a high carbohydrate diet. Here's a low carbohydrate diet. The total amount of fat oxidation is going to go way up. And the percentage, remember earlier I said like, well, the percentage varies based on training, the percentage varies based on diet and whatever. So here's a high carb diet, and here's a low carb diet. The low carb diet has a much higher VO2 max at which they can still have peak fat oxidation. So peak fat oxidation, before it starts to retract, before it starts to go down, before carbohydrates overtake the fat and your total absolute fat metabolism starts going down, for people on a high carb diet, 50-something% in this study, this is a study. This isn't the end all. But consider this a pattern. For the low carb diet, it was over 70% of their VO2 max is where they peaked in fat oxidation. And then just a few graphs here of looking at what that metabolism actually looks like. High carb diet, this is serum insulin. Here's the treadmill run, it goes down. And during recovery, insulin goes way up. For a low carb diet, it was just lower all the way through. But during exercise, it did get close. And then during recovery, it stayed down a lot. This is carbohydrate oxidation. It's what you would expect. Low carb diet, not much in the way of carbohydrate oxidation. High carb diet, tons of carbohydrate oxidation. Fat oxidation, it's the opposite. Fat oxidation during a workout, there's the low carb diet, there's a high carb diet. High carb diet, you do oxidize fat. Even though there's a little bit of insulin on board, the insulin does crash, you are able to mobilize fat. But the rate of lipolysis and the rate of oxidation are slower. And then your ketones. Hydroxybutyrate, that's the beta hydroxybutyrate. That's the metabolic one everyone's measuring. So total ketones and the metabolic one. The low carb diet has a lot more ketone metabolism, ketone activity. Tons of articles. All of these articles are on the website. You can find them. So acetoacetate and hydroxybutyrate, these two, that's what commonly what you'll see measured. Acetone, that's the one that you piss out. If you want to see if you're in ketosis, you have to pee on the strip. Acetone, that's nail polish remover. That's the one that you pee out. So you combine two acetyl-CoAs and you make your acetoacetate that way. So postprandial exercise, everybody's gonna eat big doses of carbohydrates. It's a holiday, it's a whatever. You're gonna have a big meal. You're gonna go to a buffet, you're out to eat, you're whatever it is. You're gonna eat a bunch of carbs. Postprandial exercise, meaning post-meal, postprandial, post-meal. You can use AMPK to do insulin's job for insulin, so you're not gonna get the insulin spike. So remember, AMPK is going to block glycogen synthase. AMPK is going to activate hexokinase. AMPK is going to activate phosphofructokinase. Both of those are glycolysis, so we're consuming those carbs. And then AMPK is going to mobilize GLUT4. So AMPK is a better substitute for insulin, and you will turn on AMPK through exercise if you do some sort of long bout of exercise after you're done eating all the rolls or whatever. Because if insulin doesn't, if you rely on insulin to get the sugar out of the cell, out of the blood and into the cell, you're going to activate PDE. PDE is going to block lipolysis. You're going to reduce the amount of adipose triglyceride lipase you have. So there's a chronic effect, there's an acute effect. AMPK, the chronic effect is mitochondrial biogenesis. So not only with AMPK are you avoiding the suppression of adipose triglyceride lipase, but you are increasing the amount of mitochondria you have. So AMPK is very helpful in these situations. If you want to exercise in a fasted state, there's a ton of evidence that you're going to burn more fat in that workout, even with resistance training. One of these is resistance training, this one. Resistance training, even there, you'll have more fat oxidation in that workout, short term. Long term, it depends on who the person is, how effective that's going to be.
So you have AMP activating AMPK going into mTOR.
Inhibiting mTOR, yeah, so you're inhibiting mTOR with AMPK.
Oh, okay.
Yeah.
Makes sense. Okay.
Did I have that on?
No,
Okay.
I think I just put it on.
Okay. Yeah, so through tuberin and raptor, you're inhibiting mTOR.
How does that work with PDE3?
As in you're getting a lot of AMP?
You're
Yeah.
creating AMP.
Yeah, I know, it gets messy. Yeah, I was hoping nobody would ask. It gets messy. So with PDE, you do create AMP. AMP is the trigger of AMPK. It's in monophosphate, AMP activated protein kinase. I think it's matters of magnitude. I think it's matters of scale of how much AMP are we talking here.
Okay.
And it does get a lot of messy, and there's a lot of overlap in some of these things. Yeah.
Yeah, because I'm wondering if you have insulin, you're trying to go through mTOR, then you're inhibiting catabolism, right?
Yeah.
The PKA pathway. So then, but you're also going to be inhibiting the PKB pathway.
Insulin, it's just so direct in the activation of PI3K, PKB.
Okay.
Yeah, it's just such a direct activation. It's the same thing with when I say caffeine is, or theophylline or theobromine, any of these methylxanthines are non-selective inhibitors of PDE, phosphodiesterase. So coffee, you're blocking PDE, but man, insulin's effect on it is like 20,000 times stronger.
Okay.
Insulin is so powerful.
Okay.
And so is caffeine, can you just drink a ton of caffeine and get a PDE inhibition effect? Yes. It's small. So sometimes some of these signals, it's not like they're all as loud as one another. Like you're in the choir and one person, you can hear them from like Lodi, and the other person is like, oh, they have a nice voice. I wish they weren't just muted by the person next to them. Like I can just be. So matters of magnitude, but other than that sort of vague answer, I have a hard time, quantifying things. Poor sleep. Just know that sleep, if you don't have your sleep dialed in, don't even try with anything else. I've been sleeping so badly the last couple of weeks, and so there's almost no point in doing anything in life. Sleep is so important. And so short sleep duration, we're looking at changes in leptin, changes in ghrelin, changes in testosterone. In the short term, changes in growth hormone, which are restored pretty quickly if it's just short-term sleep deprivation. Long-term sleep deprivation, we're seeing some more complications. Glucose, homeostasis, diabetes, everything that makes fat loss challenging is made worse by a poor sleep schedule. So you have to have that done. Remember, diets of exclusion. If you're avoiding sodium, if you're not putting salt on your food because of blood pressure, okay, well, maybe what we're seeing is the iodine. Like iodized salt is often the major salt might be the major source of people's iodine. And you see the same thing in vegan diets, things like selenium, things like iron, iodine. Those you commonly see deficits. So especially selenium and iodine, you see a lot of diets of exclusion having deficits in these. And so excesses don't help. Deficits make it challenging, just like sleep. If you don't have your sleep dialed in, I don't think that supplements aren't gonna help if you don't have your sleep dialed in. And so doing the tricks is not helpful if you don't have the fundamentals. And so making sure iodine, selenium, magnesium, zinc, and then iron, if you have deficiencies in any of those, that can be problematic. And just making sure you have that okay to move on. Again, excesses aren't like, oh, now I have an awesome thyroid because I ate 4 pounds of seaweed today. That's not additionally helpful. You just can't have deficiencies in those things. Remember, heat. When people do like Bikram yoga or exercise in the heat, that compromises fat metabolism. It makes it harder to burn fat. And the sort of crude explanation is shifting the oxygen dissociation curve. It's a little bit harder to deliver oxygen to the tissues. Fat metabolism is very oxidative. It's hard to deliver that. But we're seeing some other complications as well. So there's a lot of animal models. It's cows and it's pigs. That's usually what they're looking at. But the heat-stressed animals have a really hard time doing lipolysis. So non-esterified fatty acids. Here's heat-stressed, here's normal. Lipolysis gets messed up in the heat. One thing, sometimes they'll see insulin being higher in heat stress. Secretion of insulin being higher. Oxidative enzyme activity is compromised in heat stress. You mobilize more carbohydrate in the heat stress. You're much better at mobilizing carbohydrate out of the liver. But oxidative enzyme activity gets ruined by heat. And this is just looking at glycolytic activity in people. So the high heat condition, you lose a lot of glycogen before to after. The control condition, you don't lose as much. There's a lot more, and multiple studies have looked at this exact same thing. Heat stress makes it hard to lose fat. So getting in that sweat box or whatever reduces fatty acid oxidation. Coffee is at least fine, if not good, if not very good, if not best, if not even better, right? So, but caffeine, you need a big dose of caffeine for the phosphodiesterase inhibition. Pretty big dose, you need a lot of cups of caffeine to get a meaningful effect of phosphodiesterase inhibition. You need a tiny dose to get the adenosine receptor, blocking those adenosine receptors. And so that's more of the effect of caffeine is coming from adenosine antagonism as opposed to phosphodiesterase inhibition. Like theophylline, you don't need as big of a dose. Theobromine, you can eat a lot of chocolate and get that dose, I guess. But in all of these methylxanthines, these PDE inhibitors, you do need a little bit more to get that PDE inhibition dose. Where insulin, it's just like, I heard insulin whispering around the block, and PDE is just alive and active. Okay, so the supplements. All 6 of these articles are in last lecture's PDF folder. You don't have to pull them up. I'm not holding you accountable, but the supplements that just don't do anything. Okay, not they don't do anything. That's every supplement ever has an effect. There has never been a supplement. Placebos have effects. Every single. What's the most benign thing there is? I'm going to tell you it has a huge effect in the body on something.
something in mind?
I don't have anything in mind. Yeah, I'm just curious. I'm surveying for. I mean, I guess if you have like an IV bag, right? You're just hydrating yourself.
That's a huge thing.
Huge effect on metabolism. Look, read Florian Lang. Florian, Florian Lang's work. The metabolic effect of just dripping in a little hydration into you. Cell swelling, cell shrinkage, look at what those cells do in response to that. There's nothing benign in the world. Now, what effect does it have? Does it have an effect on lipolysis, beta oxidation? Does it have an effect on fat metabolism somehow, weight management? These ones don't have an effect there. They have an effect on other stuff, beta glucans. GP isn't here. Okay, Nathaniel, what do beta glucans do?
They bind to bile, and they remove cholesterol.
Yeah, so you have to remake your bile. You're gonna shit out your bile because where fiber goes, this is a soluble fiber, beta glucans is the stuff that makes oatmeal famous, and why it has his hearts on it. Binds to your bile. Bile has a bunch of cholesterol in it. You shit it out, and you have to make more bile, and like your HDLs go down too, but your total cholesterol goes down. So a lot of these, green tea extract has effects. Those polyphenols, those are good, but it doesn't seem to work for weight loss. It's not supported in the literature for weight loss. Guar gum, as a food thickener, wonderful, and I'm sure it has some effects. There are some studies that show satiety.
These are supplements on weight loss?
For weight loss, yeah. These are supplements that do not work for weight loss. They do work for things like cholesterol. But for weight loss, these supplements don't have an effect. The study after study, it's just like we didn't find anything. We tried and tried and tried, and like those claims don't hold up true. These ones have some support in the literature. I probably say the best ones on this list are berberine, yerba mate, and it's young. The research is really young, but the raspberry ketone. Raspberry ketone, this is in raspberries. If you can smell it through the mask, that's what you're smelling. And it's encouraging, but it's too young for me to induct it into the sort of Hall of Fame. Pyruvate, there's a little, it's not missing from the literature. It's the studies, there is some support. So these ones, if you're like, you know what, I'm just gonna give everything a go. Okay, you can try these ones. There is support in the literature for every single one of these. CLA, really popular. And there is some evidence for CLA. So every one of these has some evidence behind it. It's just not, like, if you're like, you know what, I just want to spend the minimal amount of money and I got to be sure this thing's going to work, skip this list. If you're like, you know what, I just need to, I'm going on Biggest Loser and I want to weigh 6 pounds by the end of October. Give it a shot. See how these things work. Cameron, you're writing frantically. Do you want me to?
Oh, no, it's okay. You can sit.
Okay. These are the ones that have more compelling support in the literature. Not that every single study ever has shown support, but in meta-analyses, we're looking at some amount of support. Capsaicin, that's the thing in peppers that makes them hot. It's one of the compounds. Capsinoid is in peppers and doesn't work. This has been studied a bunch for its claims and never turns out to work. Capsaicin has turned out to work. You know what carnitine does? That did turn out to work in the end. Psyllium is just a fiber. We cook with it all the time. It just like, man, everything gets goopy. It's just like gelatinous and goopy, but it's just a fiber. And I have never, I've done. I've never done those three, but there is support for those three in the literature too. Okay.
Not the fourth one,
Forskolin? Yeah. Bulletproof, Bulletproof Coffee, that company. I don't really like the guy because he says like really weird things all the time, and it's like that's not even close to true. Listen to him trying to talk about mTOR. But we read his book, and just like every page was like, whoa, that's false. But I like their coffee. It's my favorite coffee. So, but they released that one. There's on the previous page. There. Yeah, so they had that, like, I'll give it a shot.
So looking at like the $69 for the 60 capsules, I'm like, What's the cost of the plant? Like, just leave
Can you just grow it in your yard? And like, yeah, it's probably like an invasive species, and it'll cost you 4 cents of whatever, water and stocking's expensive. Are we okay with the supplement lists? So if you're looking for something that is a little bit more encouraging in the literature, again, I've done some of these. I've done a few of them, 5 of them or something. Like nearly all of them. Glucomannan, CLA, forskolin, everyone's calcium, whatever, berberine. I haven't done raspberry ketone, or I haven't done pyruvate from the sense of as a supplement, or mate. But I've done most of those. I've done more of the mites than I have of the do's. That's fine. But that's what the literature says.
back one to the dues.
The do's.
So the carnitine supplement, there is evidence?
There is
We're talking
evidence.
about that.
Early evidence was less compelling from what I saw. I haven't seen every single study, but at this point, all you have to do is look at meta-analyses, and they do all that heavy lifting for you. And so there's enough evidence in the meta-analyses that carnitine is supported, especially supported in particular populations. When you look at, HIV was the population that's like immediately, it's the best thing in the world. Like steroids are probably the best thing in the world for HIV-related muscle wasting, and when you get the lipodystrophy, stuff like that, carnitine, man, that's effective. But in non-pathological populations too.
Did we talk about where carnitine originated, like how it's created?
Yeah, lysine. Yeah. So it's derived from lysine. Yeah, lysine does a lot. Remember when you're looking at mTOR activation, lysine is very helpful for that too. If you want 90% of mTOR activation, leucine, lysine, arginine, you're at 90% of mTOR activation. That's also what you get carnitine from. Lysine is useful. Okay, fasting. Who has done intermittent fasting or just like on accident fasted for a long time? Like you're on the road and you forgot to bring food, or you're locked out of your house or something? Usually at least one weekday. It's either Monday, Wednesday, or Friday. I'll fast for the day. I'll do about 36 hours. I'll go to bed one night, and then I won't eat anything until like 2 days from then's breakfast. And all I'll do is have water. Occasionally, I'll put a little salt in my hand and lick it. Like hyponatremic, but if I'm like exercising, if I'm sweating, if I'm whatever. But all I'll have is water for 36 hours or something. And if you've never done it, you should. I think Van Ness has his class do it as part of it. You have your class participate.
No, I don't force them to do it. They all did. I think they tried a 2-day fast, and
I've never done it too.
most loved having done it.
Yeah, that's funny. That's a good way of putting it. I love having, but man, you love food the next day. Really not at breakfast. At breakfast, I'm like, I could go either way. I can keep fasting. But after a couple of hours into the morning, nothing has ever tasted so good. You really sensitize your palate and your appreciation for food if you deny yourself from food. But you do the same thing with like insulin receptors. The insulin has never tasted so good, says your insulin receptors. There's a lot of evidence, and we'll sort of hasten our way through it, and we'll recover this stuff on Monday, whatever. But just looking at fasting, this article is in lecture, I think it's 7's PDF. We've used this article before to talk about lipolysis. In the first lipolysis lecture, this article appears. I'm pretty sure it's lecture 7, that PDF one. We're just talking about the regulation of lipolysis, adipose triglyceride lipase, hormone-sensitive lipase, catecholamines, right, epi, norepi, activating PKA. There's a nice explanation of this stuff, the basic process of lipolysis. Great article. But when we fast, what happens to that stuff? What happens to lipolysis when we fast? And fasting, just a very general, what do I mean by fasting? Or different articles, different people, different coaches have different definitions of fasting. Different populations will use that word fasting differently. So periods that range from half a day to 3 weeks. That's a broad definition of fasting.
Isn't 3 weeks like that?
If you're like too dehydrated or something, but I mean, the longest fasts are pretty, I should look it up. But the longest fasts, those are long. There's no way I'm
40
doing it.
days.
Pardon?
Jesus, 40 days.
40 days in the desert. So yeah, that's, and you've seen the pictures on the cross. That guy had ripped abs. Every painting is just veiny ripped abs. So that's what that does for you. You super lipolysis. Those are accurate from that perspective. So I've highlighted passages that I think are important. But George Carlin had a really nice line when he was alive about when people say, I am starving to death. That's what starving means. Starving is not voluntary. Starving is not endured temporarily. Starving ends in death, or it at least is delivering you to death. So fasting and starvation, not even close to the same thing. Starvation is involuntary. Starvation is symptomatic and potentially lethal, and you don't want to be starving. So that's what starving means, is to death. That's implied by the word. So fasting is a deliberate withholding of food or restriction of any calories. It is not the same thing as reduced caloric intake. Fasting and like, oh, just eat 200 calories a day. That's not fasting. Now, there are things like protein fasts and fasting-like diets where you can get some of these effects without completely denying yourself anything to chew on. There are diets that can mimic some of this effect. You can sort of eat some fat, really. But if you deny yourself protein and deny yourself carbohydrate, you can elicit some of these effects. And so that's just what these first few things are, is the definition of what do we mean by fasting. This one, we now know that fasting results in ketogenesis, promotes potent changes in metabolic pathways and cellular processes such as stress resistance, lipolysis, autophagy. Let's break down our proteins and have medical applications that in some cases, it depends on the cases, can be as effective as drugs. Depends on the cases. And fasting in combination with drugs, nutritional interventions in combination with pharmacological interventions seems to complement each other. Maybe just the fasting on its own is not doing the trick for somebody who has a tumor now, but fasting or fasting-like diets in combination with the treatments, with the medical treatments, is much more effective than the medical treatments on their own. Same article right here. All these articles will be up later today. After hepatic glycogen depletion, doesn't take that long. Doesn't take maybe a day if you're fasting. If you're running a marathon, you're going to be out by the end of the marathon. Right? Your glycogen is gone, and you see this in the runners. They don't know where they are. Right? Somebody has their arm around and they're escorting them across the finish line, and they just look. You see it in their eyes. That is an insane person who just needs a squirt of orange juice or something. But the ketone body's fat-derived glycerol. Remind me what glycerol is. It's the backbone of a triglyceride, a triacyl glycerol. And remember, that has its own fates. When you free that up, that thing has its own metabolic fates. You can make carbs out of it as an intermediate in glycolysis. And amino acids, so those things and amino acids account for the gluconeogenesis dependent generation of about 80 grams a day of glucose. Your brain is still using glucose. During fasting, your brain is like, oh, you know what, me too. Yeah, I'm not gonna eat anything either. That's not what your brain does. Your brain still needs the carbs. And so one assumption that just seems intuitive would be like, well, my brain isn't going to work that well when I'm fasting. It needs a bunch of carbs and it needs a bunch of calories and I'm not giving myself anything. My brain must not be working. That's not how it works. That might be intuitive, but that's not how it works. So during prolonged fasting, fed versus starvation or prolonged fasting conditions, what we see is glucose. Glucose isn't providing the bulk of your calories. Your ketones, acetoacetic acid, and then into beta hydroxybutyric acid or butyrate, hydroxybutyrate. So that's the bulk of it. That is the bulk of your calories is ketones during prolonged fasting. And this depends on the person. It depends on the person, how quickly you get here, how many days does it take to get there? What was your diet like before? What's your insulin sensitivity? What's your history of training? How old are you? All of this matters. So this isn't just like every single person ever looks like this, but this is a snapshot of general expectation of what you would expect to see. So we talked a lot about diabetes and metabolic syndrome. Metabolic syndrome, right, we're seeing abdominal adiposity, right, we're seeing compromised cholesterol, lipid panels, we're seeing elevated blood pressure, right, we're seeing insulin sensitivity problems and elevated blood glucose, we're seeing all these things. Fasting seems to address them all. MS, that's not multiple sclerosis, that's metabolic syndrome. And so all of these things seem to be improved by fasting, right? We're saying, and there's a lot of like in rodents, animal models, human models too. Abdominal fat, right? So metabolic syndrome, that's one of them, that waistline. Inflammatory profiles, blood pressure, insulin sensitivity. All of these things are improved in the presence of a fasting protocol. And there's a bunch of hormones. The hormonal changes that typify metabolic syndrome. We reverse all of this by fasting. I'll show you all the specific stuff, but leptin levels, adiponectin levels, ghrelin levels, insulin levels, all of these, when we see metabolic syndrome, the leptin profile, the ghrelin profile, right? When we see insulin levels and insulin sensitivity and adiponectin, remember that's something that fat cells release and it promotes insulin sensitivity. If you go exercise, don't just go like fill your mouth with food immediately afterward if you're looking at improving this stuff, because you can abolish some of that adiponectin effect. You reverse all of those hormone profiles, what those look like. And remember, fasting literature is younger than the cardiovascular response to exercise. And the cardiovascular response to exercise, responders, non-responders. HDLs can go down with exercise, right? In a percentage of people, you worsen your HDLs with aerobic exercise. In a percentage of patients, you increase your systolic pressure. In a percentage of patients, you increase your triglycerides, right? That Claude Bouchard paper, where he just looked at the individual, not the means, not 4 to 9 millimeters of mercury, but the individual response, and he found a percentage of people who had a terrible response to exercise in almost every cardiovascular parameter. Fasting, we're maybe not there yet in terms of understanding responders and non-responders. So there's some generalities, there's some means, there's some animal models we're extrapolating from. But fasting in the brain, when you're denied food, this is the time your body decides, I need to be able to acquire food. What I was doing before wasn't working. I haven't eaten in 24 hours. Lounging around or looking around, that wasn't working for food acquisition. I need to be a little bit more vigilant. I need to be a little bit more creative, maybe some lateral thinking or just like more attention. You get it with fasting. Case by case basis, I get it. If I do fasting too often, I sort of get used to it. And I don't get that effect, and I don't fast for a little while, I do it again, and then I become attentive. It's almost like a mild Adderall effect, where you get this sort of hyper focus. But that's seen in the literature, and you get BDNF, brain-derived neurotrophic factor. That's about the best thing you can put in your brain. It binds to TrkB, tropomyosin receptor kinase B. It's just a receptor. You have TrkA is what nerve growth factor binds to. If you don't have TrkA, you have SEPA. Does anybody know what SEPA is?
The pain. You don't feel pain.
You don't feel pain, congenital insensitivity to pain, and anhidrosis, SEPA. If you can't have nerve growth factor binding to TrkA, like goodbye nerves, and TrkB is what BDNF, brain-derived neurotrophic factor, comes from. And in fasting, we see increases in BDNF. There are supplements that seem to increase BDNF. The coffee berry has some support behind it. Not coffee, but the berry that isn't in the coffee. There are things, exercise increases it a lot. We're gonna end with a very similar slide, 10 minutes or whatever, but people have started looking at all of these different effects of fasting, whether it's at the skeletal muscle, whether it's at the adipocyte, it's the intestines, it's the brain, the liver, the heart. And again, the research is relatively young. This is 2014, but so many effects are being observed, and most of them are positive. Most of them are positive, but some of these early effects, for the muscle, increased insulin sensitivity, enhanced anabolic signaling. That one, 2014, we sort of are developing a different story today of what that looks like. And the story is growth hormone. The story is a little bit more wordy than enhanced anabolic signaling though. But increased stress resistance, reduced body temperature, I don't know, it's not necessarily perfect, but what we see is, here's the growth hormone. Now, you fast, you're hungry, okay, your stomach is empty, and it's just like little and tiny and sad. Your stomach is sad, okay? A sad stomach releases ghrelin, gastrointestinal tract, stomach, releases ghrelin. Ghrelin is spelled G-H-R-L-N. Growth hormone releasing, G-H-R-L-N, ghrelin, growth hormone releasing. Ghrelin released from the stomach in response to a prolonged absence of food stimulates the release of growth hormone. Growth hormone enhances lipolysis greatly. Most of the tissue in it, because we think of growth hormone, because people take growth hormone, and you're like Sylvester Stallone, or you're Ronnie Coleman, or whatever, and like you're huge. Anabolism, you're going to get huge with growth hormone. The problem with talking about fasting and anabolic signaling is growth hormone doesn't really do it directly. Growth hormone doesn't really make your muscle huge directly. It's the boss of IGF, insulin-like growth factor 1, IGF-1. It takes some hours, you get a big growth hormone surge, and then later you get IGF coming out of the liver. Hypothalamic pituitary liver axis. You're going to get IGF. IGF does the heavy lifting. IGF, it's a negative feedback loop, is going to inhibit growth hormone, IGF-1. What we see in fasting is not the same thing you see when you get other surges of growth hormone. You actually get a suppression of IGF in tandem with growth hormone. And so in part, IGF would be inhibiting growth hormone, and maybe that's part of the signaling of we don't have that backward inhibition of growth hormone release, but also we see binding protein 1, IGF-BP1. There are 6 binding proteins for IGF. If IGF is bound to a binding protein, it can't activate its receptor. You are inactivating IGF by binding it to a binding protein. 1 is not really a common one. 98% approximately of your IGF is bound to binding proteins. Binding protein 3. When you make IGF in the liver, you're releasing binding protein 3 too. And that's about 80% of all circulating IGF is bound to binding protein 3. Binding protein 1 though, you're increasing this. And so the IGF that you do have is inactivated. And so you actually get less anabolic signaling. And it makes sense. It doesn't make sense when you're like, I got more growth hormone, I should be growing. But it makes sense from the perspective of starvation or food deprivation or fasting or any of these kind of long-term kind of removals of caloric signaling. If you did have growth hormone with the IGF, that would be horrible for cancer. So if you fast and you get that canonical signaling of the hypothalamic pituitary liver axis to get the IGF, fasting would be like the worst thing you could do for cancer. So growth hormone itself, this doesn't seem to be harmful for cancer. It doesn't seem to accelerate tumor growth or promote metastases. IGF does, but you get growth hormone without the IGF. So you don't get the anabolism, which is great for things like cancer. But you do get all the effects of growth hormone that really aren't that. Accelerated lipolysis is one of them. Just looking at what the IGF signaling cascade would be doing. So this is a 2018 article, really starting to get into this. Now we'll talk all about IGF signaling up next. We're going to get into anabolism, mTOR anabolism. But IGF would be doing essentially like insulin signaling. That PKB, AKT is PKB. So that PKB, you're going to inhibit if you're doing IGF. Fork head box O. This is like cancer regulation stuff, and IGF shuts it off. At the same time, it turns on cell growth. So IGF gets suppressed in fasting. But this is just notably standalone therapies, periodic fasting or these sort of fasting mimicking diets. Ketogenic is sometimes lumped into these. There's limited efficacy against, if you have an established tumor, but used with medications, it seems to work way better. You're really enhancing the medical treatment with these diets. Now, a lot of this, when you look at things like AMPK, great for cancer prevention. It's a little bit trickier. Once you have cancer, it depends. Is it protecting the cancer? Or is it inhibiting metastases? It depends on the type of cancer. It depends on the patient. Preventing cancer, those things are very helpful. So getting into fasting and exercise, looking at endurance performance is not helpful if you're performing. There's one study, this is a 2020 article that was looking at just polling, just sort of a state of evidence, sort of a review of what we know now. And there's one study that showed this very modest effect of improved performance. Most studies, then there's more than they're citing here. Most studies say if you're fasting, don't fast on race day. That's not helpful. If it's a race day, don't go fasting. And this is the one where, well, actually this one was I think also, they didn't perform well, but people who are in better shape, fitness, aerobic fitness, right? Those people seem to resist decrements in performance if you come in better shape. If you're if you finish the shuttle run last, or you're doing like the mile run and you come in last place, fasting may be more compromising to your performance than the person who finished first. Protein metabolism. It's mixed evidence. Protein metabolism is mixed evidence. Some studies have found it doesn't affect protein metabolism. Short-term fasts, long-term fasts are going to affect it. Short-term fasts, 24 to 48 hours, something like that. There's some studies that found that it does not affect protein metabolism. Other studies have found that it does. Other studies have found even intermittent fasting, like Ramadan, where it's I'm going to no eating in the day and I'm going to eat at night. That some of those studies have found increased catabolism of protein. So mixed evidence, it's not increasing anabolism. Mixed evidence on how compromising it is, if it is compromising, and to what population. So just a few things here. So exercising during fasting. If you're looking at lipolysis, you want to increase lipolysis, and you want to get good effects of fasting, that's helpful. You're not going to be at your best, right? Again, don't do it on a race day. There's implications for train low, compete high, train while fasted, and then feast and compete. Okay, maybe. I haven't seen the evidence necessarily for that, but physiologically, that makes sense. But in terms of fat mobilization, fat oxidation, training while fasting does work better. Your phosphorylation of adipose triglyceride lipase and hormone-sensitive lipase, so increased lipolysis, fatty acid transport, upregulation of the genes responsible, fatty acid translocase, right? You know what that is, carnitine, these enzymes to help shuttle these proteins that shuttle your fats in, get you ready for beta oxidation. So you're going to mobilize more fat. You're going to be able to get that fat where it belongs, and then you see the effect of more oxidation in a fasted state than you would get in a fed state, even after the workout. If you eat, feeding before you'll increase insulin levels, it's not much if you're going to exercise. You'll deplete some of that, but there is still an effect by way of comparison. And so you will be much more lipolytically active in a fasted state. And so the last one, this is just like the last thing, it's just there's a ton of stuff. This article is in there, and it's New England Journal of Medicine. Mark Mattson is one of the people I cite a lot. He's talked a bunch about like sulforaphane, a huge researcher, Mark Mattson. But the New England Journal of Medicine is very comprehensive in what it's looking at in all of these different functions, systemic functions of fasting. Performance is not helpful. Protein metabolism from the perspective of growth is not helpful. Pretty much everything else is very helpful. We'll go over this again in a little bit more detail, but periods of intermittent fasting, what the recovery looks like, long-term adaptations, insulin sensitivity, heart rate variability. I think that's what HRV. What else would HRV be other than heart rate variability? Maybe it's heart rate variability. Lipid metabolism, the gut microbiome seems to improve, reduced abdominal fat, reduced inflammation, reduced blood pressure. Long term, there's a lot of good effects. And again, this is another Mark Mattson one, just looking systemically. So many perks, human performance and protein metabolism from the perspective of growth. Those are not perks, but everything else seems to be brain and elsewhere. Okay, that's it.
Heat recovery ventilation. Another possibility.
What are you looking at?
HRV.
Oh, what is it?
Heat recovery ventilation is another option. But
Okay, that's a heat recovery ventilation. That's the one, not heart rate variability. It's an HRV. Yeah, isn't HRV like some sort of, that's a Honda? Yeah.
CRV.
It's the CRVs, yeah. Oh, 9.
There's a guy in Sri Lanka claiming to be a breatharian, which is says it's only breathed air, hardly eaten during the past 5 years before we might be feeling wind and
I love that. You gotta hug me while you say that.
How far into the fast do you have to get the fat oxidation?